Good afternoon, everyone. I'm Dr. Warunie from the Department of Anatomy. Today we are going to learn about muscular tissue. If you have any questions or need any clarifications, you can disturb me at any time. Okay. So these are the things we are going to discuss today about the types of muscle, skeletal, then each of the types separately, the skeletal muscle, about the organization, the contraction, innervation, and the types. Then about cardiac muscle, again the organization, contraction, and a bit about the conduction system. And then the smooth muscles, the organization, contraction, and innervation. And last, we'll discuss a bit about the regeneration of muscle tissue. So muscle tissues are cells optimized for contractility. So they are specialized for contraction. And the three types are skeletal muscle, cardiac muscle, and smooth muscle. And the contraction of these muscles are caused by two types of filaments, thick myosin filaments and thin actin filaments. We'll see about them in the later slides. Okay. So if we look at a muscle cell, the structures are called different names. So if I draw a usual normal cell, there are some structures like this, right? So can someone tell me what is this? This outer line?
Plasma membrane.
Plasma membrane. So in skeletal muscle, because they are specialized for contraction and different from the typical cell, they are called sarcolemma. Then this area, yes?
Cytoplasm.
Cytoplasm. So in the muscle cells, it's called sarcoplasm. And these are, if you can identify, what's this?
Mitochondria.
Mitochondria, yes. So in the muscle cells, they are called sarcosomes. And what is this?
Endoplasmic reticulum.
Yes, endoplasmic reticulum. And in the muscle cells, they are called sarcoplasmic reticulum. So then you have the nucleus. It doesn't have a special name. So when we look at the skeletal muscles, they are also called striated muscle fibers. So these muscle fibers have lines. So they are called, so because of these striations, they are called striated muscle fibers. They are long. Sometimes they can span the whole length of a muscle, right? So if this is a muscle, these cells can span the whole length. Some can be short and some can span the whole length. Right? They are long and cylindrical. So they are like cylindrical in shape, right? And one cell has a lot of nuclei, right? And these nuclei are elongated and are found at the periphery of the cells. Right? The diameter of these cells, the diameter can range from 10 to 100 micrometers. And the contractions are strong, quick, and voluntary. Right? They are controlled by conscious control. Right? Then if we look at the organization of the skeletal muscle, these muscle fibers are covered by connective tissue in an organized manner. Right? And within these connective tissue, you find nerves, blood vessels, and lymphatic tissue. And they also help to transmit forces. Right? Contraction forces are transmitted from one muscle fiber area to other nearby areas through this connective tissue. So if we look at the organization, a whole muscle fiber, the muscle fiber is covered by the epimysium. Okay? So this is the epimysium which covers the whole muscle. Right? Then we have muscle fibers together. These areas are called fascicles, right? These muscle groups are called fascicles, and they are covered by the perimysium. This, if I use another color, this connective tissue is called the perimysium. And then when you take one fascicle, you have many muscle fibers. So these are muscle fibers. Right? So these muscle fibers, see, the single muscle fibers, they are again covered by connective tissue. And these connective tissue are called endomysium. So when we take one muscle fiber, one muscle cell, each of them have many myofibrils, right? So these are myofibrils. So one muscle cell can have about 1,000 myofibrils, right? So here we have the structure of one myofibril. They are very well organized and have the contractile proteins which are actin and myosin. Right? They have recurring units which are called sarcomeres. Right? So these recurring units which are called sarcomeres, when we look at them, they are arranged like this. Right? You have actin, the protein actin and myosin. So actin are the thin filaments, right? You can see they are thin. Then here, the myosin fibers, they are thick, right? They are also called thick fibers. So if we look at that structure more precisely, here these are the actin filaments. And these are the myosin filaments, right? And to describe this structure, the actin filaments, they are attached to a disc. This disc is called the Z disc. And the thick filaments, they are attached to a disc which is called the M line. Right? So these areas can again be divided into bands. There are the I bands and the A bands. The I bands are more light colored, right? So I bands are light colored because they only have the thin filaments. The area covered by only the thin filaments, right? Then when you take the A bands, they span the length of the thick filaments and they are more dark staining. Right? So this is an electron microscopic view of the same. So here you can see the Z discs to which the thin filaments are attached. Right? So a sarcomere is described as the space between the Z discs. Right? Then you have the M line to which thick filaments are attached. Right? Then you have the bands, the I band, which has only the thin filaments, and then the A bands, which have only the thick filament, which have spanned the length of the thick filaments. Right? Then again, you have a zone called the H zone, which span the length where you find only the thick filaments, right? This area, you find only the thick filament, right? Okay. So you can see the colors vary accordingly, right? So here it's light colored because it only has the light, only the thin filaments. Then here in the A band, you have the thick filaments. In this area, you have both filaments overlapping. So it's very dark colored. In the H zone, you have only the thick filaments. Okay. So this is the light microscopic view of the skeletal fibers, right? You can see the striations, the lines, the I bands and the A bands. So I bands are light colored, A bands are dark colored. So this is a nuclei. You can see that it is situated peripherally. And this is a fibroblast. So this is also the same. You can see here the different myofibrils. They are not in the same line. They can differ a bit, right? Okay, so now we will look at each of the fibers. Okay, first, we have the myosin filaments or the thick filaments. They are large complexes weighing about 500 kilodaltons, right? So if you take one thick filament, it has many myosin molecules, right? So when you take one myosin molecule, it has a heavy chain. The heavy chain has a tail. This is the tail of the heavy chain, and this is the head of the heavy chain. And it also has myosin chains in this area, in the tail and the head. So there are two heavy chains and two light chains in each of the molecules. So this, when you take the myosin head, it has two binding sites. One is for actin, the light chains, right? And the other is for ATP. So ATP is required for the energy for contraction. So there are several hundreds of these myosin molecules in one of these thick filaments. Right? Then we come to the thin filaments. Right? So this is the thin filament. It is thin and helical. And it consists of these globular structures, right? So one of these globular structures is called G-actin or globular actin. And they are formed as a chain, right? So they are called, this chain is called F-actin. F is for filaments, right? Filament actin. Then these actin filaments are related with two more types of molecules, which are called tropomyosin. This green structure is called tropomyosin, and this blue structure is called troponin. So when you take the tropomyosin, it covers the actin strands, right? So you can see there are these dark spaces. The myosin heads are supposed to bind to these areas, right? Here also you can see. So these areas are covered by this tropomyosin, which is formed as a helix around the actin filament. Then you have the troponin. Troponin, there are three units. They are troponin T, C, and I. Right? So they are attached at regular intervals on the tropomyosin molecules. So this troponin T, so this is troponin T, it is attached to the tropomyosin. And this is troponin C, which attaches to calcium. And then troponin I is an intermediate structure and regulates the actin-myosin interaction. And then when we come to the sarcoplasmic reticulum, and it is organized with the transverse tubular system. Okay, so we will look at a picture. So, yeah, this is the T-tubule system. Right? So this is the plasma membrane or the sarcolemma, right? So from the sarcolemma, the plasma membrane is invaginated into the sarcoplasm or the cytoplasm, right? So the plasma membrane goes in like this. So these lines are usually found around the Z lines. And these T tubules, you can find sarcoplasmic reticulum on either side. So sarcoplasmic reticulum are organized on either side. So when impulse is, when an impulse comes from the nerve to the plasma membrane, the message is carried out through these T tubules very quickly, right? In an organized manner, so it can be spread throughout the muscle quickly. Right? So then, because of this arrangement with the sarcoplasmic reticulum on either side, there is quick release of calcium, which is needed for the contraction of the filaments. Right? Yeah, so you have the sarcoplasmic reticulum, which is the endoplasmic reticulum of the muscle. And in this sarcoplasmic reticulum, you have a lot of calcium. Right? And this calcium is released by motor nerve impulses. So for the uniform contraction of all myofibrils together, you have the transverse or the T-tubule system. Right? So we said that they are invaginations of the cell membrane and penetrate deep into the sarcoplasm and cover each of the myofibrils. Right? So on either side of the T-tubules, you have expanded sarcoplasmic reticulum. These are called terminal cisterns. So then we will look at the mechanism of contraction. Right? So when there is an impulse, there is calcium released into the cytoplasm or the sarcoplasm, right? Then this calcium comes and binds to troponin C. Then there's a change in this troponin structure, and it helps to move the tropomyosin away from the actin binding sites, right? So these dark areas, which are supposed to bind the myosin heads, they are uncovered, right? So when they are not covered, the myosin heads, these heads, come and bind to the actin filaments, right? Then for the contraction, you need ATP. So ATP also comes and binds to these heads, and then it stimulates this head to contract or to bend forward like this. Right? So when it bends forward, the ATP is broken and released as ADP and a phosphate group. And this active filament, moves this way, increasing the overlap. Right? So this happens continuously. This binds to this, and it moves a bit inward. And then again, if there are calcium and ATP still present, it again binds to somewhere here, and again pulls it inside. So gradually, this actin filament moves to this direction and increases the overlap. Right? You can see the light chains are well overlapped on the thick chains. Is it clear for you?
Yes, madam.
Okay, you can ask me any questions if you need any time. So I have just summarized what happens, right? That action potential arises at the neuromuscular junction. Then it is transmitted along the T tubules, which are continuations of the plasma membrane. And then it reaches the sarcoplasmic reticulum on either side of the T tubules, and this triggers calcium release. So this calcium binds to troponin, and this exposes the myosin binding sites of the tropomyosin. So then active myosin cross bridges are formed, and the thin filaments are pulled into the A bands on the thick filaments. Energy is taken from ATP. So then when it is pulled, ATP is broken down and it binds another ATP, and this occurs as a cycle. Right? There are repeated cycles occurring, and for one single contraction, it requires about 100 of each of these cycles. So when the neural impulse stops, the calcium is returned into the sarcoplasmic reticulum. So when there's no calcium, again, the tropomyosin covers the myosin binding sites on actin filaments. So then the filaments passively move back to their original positions, to their relaxed length. Okay, so that's about the contraction of the skeletal muscle. Then if you look at the innervation, you have myelinated motor nerves reaching the perimysium, and each of these nerves give out branches, right? They give out several branches. These branches are not myelinated. Do you know the difference between myelinated and unmyelinated nerves?
Yes, madam.
Okay, can someone tell me what's the idea you have?
In myelinated nerves, you have concentric layers of the plasma membrane of Schwann cells. It acts as an insulator and nodes of Ranvier where the nerve impulses are jumped from one place to another.
Yes, very good. So in myelinated fibers, this is a Schwann cell, they are wrapped around like this, right? So they are, so these areas are insulated, and the impulses skip over these areas, and the impulse travels very fast. Okay, so yeah, so these individual branches travel through the endomysium and synapse with individual muscle fibers. So this is the neuromuscular junction or the motor end plate. Okay. So you can see here, there are a lot of mitochondria in the terminal area of the axon, right? And there are a lot of synaptic vesicles. So these vesicles are filled with the neurotransmitter. The neurotransmitter found here is acetylcholine, right? And when you look at the sarcoplasm, sarcolemma, that is the cell membrane, here also there are folds, right? It is the surface area is increased through these folds. Yeah. So it's the structure between the axon terminal and the muscle cell surface. You find a lot of mitochondria and synaptic vesicles filled with acetylcholine. And the area between the axon and the muscle, this small area is called the synaptic cleft. And as you saw, there are numerous deep junctional folds, and this provided a greater postsynaptic surface area. Okay. So if we look at what happens at the synapse, yeah. So you have the axon terminal, and these are the vesicles with acetylcholine. So the nerve impulses reach the axon terminal, and these move to the terminal end and release acetylcholine. So here you have the muscle cell surface, and here you have receptors, acetylcholine receptors. So this binds to these receptors, and from this impulse, it stimulates the sarcoplasmic reticulum to release calcium. So that's how the contraction starts. And this acetylcholine, what happens to this acetylcholine? Does anyone know? There's an enzyme in this area.
Acetylcholine esterase.
Yes, very good. So you have an enzyme called acetylcholine esterase. It quickly breaks down these acetylcholine molecules. So it prevents continuous contraction, right? So then the broken down acetylcholine is returned to the presynaptic surface and taken up again by the axon terminal. Yeah, so the nerve action potential, when it reaches the axon terminal, acetylcholine is released from the synaptic vesicles, and it crosses the synaptic cleft and binds to receptors on the folded sarcolemma. Yeah, and there's an influx of sodium causing deeper polarization of the sarcolemma, and this leads to the muscle action potential, which is release of calcium from the smooth endoplasmic reticulum, leading to the contraction between the actin and myosin filaments. So here, the acetylcholine dissociates from the receptor and is removed by the acetylcholine esterase enzyme. So when you take one neuron can innervate a single muscle fiber or many muscle fibers. So innervation of a single muscle fiber by a single neuron is found in places where there should be precise movements, such as in eye movements. So the muscles of the eye are supplied by a single muscle is supplied by a single neuron. But in places where there is coarse movements, like in the limbs, many muscle fibers are supplied by one neuron. So this unit, the neuron and all the muscle fibers it supplies is called a motor unit. Right? Okay. So this is a related clinical correlation. So you said that there are acetylcholine receptors which bind the acetylcholine to stimulate the muscle contraction, right? So in this disease called myasthenia gravis, these receptors are blocked by antibodies, right? Blocked and it can be destroyed, right? So these faults also reduce in this disease, and there is reduced transmission. In this disease, there is these muscles fatigue easily, right? You find it difficult to move the muscles, right? So, for example, these patients find it difficult to close their eyes and move their hands like that. Yeah. Have you heard about this disease, myasthenia gravis? Anyone? Okay, it's fine. Then the muscle fibers inside the skeletal muscles, there are of 3 types. Right? They are not all the same. They differ in their contraction ability and whether they use aerobic respiration or anaerobic respiration. So when you take type 1 muscle fibers, they are slow and oxidative, right? So these have aerobic respiration and are used for contractions which are slow and prolonged, right? Such as the muscles used for the posture. To maintain the posture, you need the muscles to be contracted for prolonged times, such as in back muscles. You don't really think about it, but they are contracted to maintain the posture. So these need a lot of energy. So it uses aerobic respiration. There are a lot of mitochondria and myoglobin in these muscle fibers, right? Then on the other end, you have fast glycolytic fibers. They have fast fibers and they use anaerobic respiration. they don't need a lot of energy for prolonged times. Their action is quick and not prolonged. And they fatigue quickly, get tired quickly, right? Such as in extraocular muscles. Those are the muscles used to move the eyeball, right? So then in between, you have type 2A. So this is type 2B, this is type 2A. These are, they are also considered fast, and they use both oxidative and glycolytic. So there's aerobic and anaerobic respiration. They are fast, they have fast contraction and show intermediate fatigue. So these types of fibers are mostly found in leg muscles. So when you take one muscle, it doesn't have only one type of these muscle fibers, right? It's a mixture. So you can see here, the different muscles are typed, are shown in different colors, right? So these light colored ones are, sorry. These are the fast fibers, which use anaerobic respiration, right? They are in white color. And the slow fibers, they are seen in dark color. And you can see intermediate colored fibers. So when you take one muscle, you can see a combination of these different types of muscles. Okay, so then we come to the cardiac muscle. Cardiac muscle is the heart, right? Here you have aligned cells which are branching. So one cell you find like this. It's also cylindrical, and the striation pattern is mostly similar to the skeletal muscles. You find the striations like this. There are 1 or 2 nuclei, and they are situated more centrally. And the diameter is, there's not a high range, it is around 10 to 20 micrometers. And in between the muscle cells, so this is another cell, you find intercalated discs. They are joined together by a special disc area. These are called intercalated discs. So these are interdigitating, right, like this. So these muscles have strong, intrinsic, spontaneous, and involuntary contractions, and the contractions, of course, they are continuous. So here you see a picture of the cardiac muscle cells. Okay? So here, these are the intercalated discs, right? There are 1 or 2 nuclei and a lot of mitochondria. They have a lot of mitochondria, and this is how the intercalated discs are seen, right? So it's not a smooth surface. There are a lot of interdigitations like this. And in these flat areas, you find a lot of gap junctions. And through these gap junctions, the electric impulses are carried from one cell to the other. So these small dots, they are the places where T-tubules start, right? So from here you have the invaginations of the plasma membrane. So this here, this is the light microscopic view of the cardiac muscle. Okay? Here you have the intercalated disc. the nuclei. And you can see the striations. Right? This is the electron microscopic view. So you can see they are, it's not a smooth surface. It has a lot of interdigitations. So this is a link for heart cells in a petri dish. You can watch that later. Right? So when cardiac cells are brought in a petri dish, they contract by themselves. So when you look at the contractile system of the cardiac muscle, it is similar to that of the skeletal muscle with a few differences. The T-tubule system and the sarcoplasmic reticulum are not as regularly arranged as in the skeletal system. Okay, the T-tubules are more in number and larger. The sarcoplasmic reticulum is less. And from the sarcoplasmic reticulum, there's a slow leakage of calcium. So this helps to recover from the previous contraction quickly, which allows the next contraction automatically. So these cardiac muscle cells have a lot of mitochondria, and this mitochondria is found in about 40% of the cytoplasm, but in skeletal muscle, it is found only in about 2% of the cytoplasm. And as these cardiac muscles need a lot of energy, they also have the lipid droplets carrying triglyceride and a lot of glycogen granules, which provide energy for the continuous contraction. So these both are electron microscopic views, right? So here, these are mitochondria. And here you can find the sarcoplasmic reticulum. And here, and these are the T-tubules and the sarcoplasmic reticulum situated close together. Here, these are glycogen granules. And this is the nucleus. This is the nucleus. Okay, so when you take the heart, there is a conduction system, right? So the contraction of the heart begins in the heart itself, right? So it starts at this place called the sinoatrial or the SA node. It's called the pacemaker of the heart. It controls all the contraction of the heart, right? So if we just look at the pathway, so the contraction starts here and the impulse travels through the atrioventricular node. So the impulse starting from here, it goes through the atria and causes the atria to contract. Meanwhile, the impulse goes to the atrioventricular node, and through the atrioventricular node, it goes into the ventricles, from the atria into the ventricles. So then you have the bundle of keys, then the bundle branches, the right bundle branch and the left bundle branch, and finally you have the Purkinje fibers. Okay. So this system, these cells you find in the SA node, the AV node, and these fibers, the bundle of keys, all these are specialized cardiac muscle cells. Okay? They are not nerve fibers. They are specialized muscle cells. But you can find a bit differences. Right? So the nodes, the AV and the SA nodes, they are smaller in size and they have fewer myofibrils. So their main function is not contraction. It is the initiation of the impulse. Right? So they don't need a lot of myofibrils. And they have fewer typical intercalated discs. They are also specialized for the transmission of the impulses. Then the fibers, they are found in the subendocardial region. So subendocardial means just beneath the surface of the heart. This is the subendocardial region. So they are pale staining, that is less mitochondria and contractile elements. Yes, fewer myofibrils. They are larger and they have a lot of glycogen for the transmission of the impulses. So these are the microscopic views, right? So here you find the SA node. So here we find the SA node. Here you find the Purkinje fibers. You can see that they are lighter staining, right? Okay, so we come to the smooth muscles. Smooth muscles are a bit different. So they are elongated and tapering like this. They don't have striations and their nuclei are single, long and found at the center. And so they are usually found in groups. So usually the thick area of one smooth muscle is closer to the ends, the thin areas of the adjacent smooth muscle cells. So they are not striated. They don't have striations. And these are linked by gap junctions. Their contractions are slow, steady, and involuntary. So they are rhythmic and wavy. Can someone tell me where do you find smooth muscles in the body?
Capillary wall.
Sorry?
Capillary wall. Digestive tract.
Yes, the digestive tract. And yeah, in the glands and what else?
Blood vessels.
Yeah, exactly. In the blood vessels you find it. And in the reproductive system, in the uterus, in the fallopian tubes, also you find smooth muscles. Okay. So here, the contractile mechanism is a bit different. There are dense bodies which can be found in the cell membrane as well as deep in the cytoplasm. Right? So there are dense bodies like this, and these are anchoring points for the filaments. Right? You have filaments extending like this between these points like this. Okay? So they are arranged in a crisscross pattern across the cell. So yeah, here you can see when these muscles contract, it becomes more globular shaped. So the other muscles, the skeleton and the cardiac muscles, just got shorter, right? But here they change their shape, right? They become more rounded. So these also, they have the actin and the myosin filaments, but the arrangement is very different. Okay, so you can see here, these are smooth muscles. And here, can you see? These are contracted, right? Their shape is a bit deformed, right? Even the nuclei, they look bent. So these muscles are attached together. These dense bodies, through these dense bodies, adjacent muscles are attached together. So they act as a unit. A group of muscles act as a unit. Okay, then when we consider the innervation, first we have known that the smooth muscles usually generate their own contractions, right? Not their contractions don't start from the nerves, they start on their own, and they can be stimulated by stretch. And as I said, the contractions travel from cell to cell through gap junctions. But they are richly innervated by the autonomic nervous system, right? Mainly the parasympathetic system. It is innervated, and they increase or decrease the level of contraction. The speed and the strength can be controlled by the autonomic nervous system, and in some cases by hormones. But in some locations, autonomic nervous system is the main thing controlling their movement rather than just modulating their spontaneous activity. So an example for this is the iris of the eye. So the pupil of the eye becomes smaller or larger by the autonomic nervous system. This happens according to the nervous system, not because of spontaneous contraction. But in the gut and glands, those areas, there is spontaneous contraction. So here, the spontaneous contracting areas, they are called single unit smooth muscle cells. Okay? They act as a single unit, but here, these, like in the eye, each of the muscles are innervated separately, and they are controlled mainly by the nervous system. Okay, so we come to the regeneration of muscle tissue. So when we talk about the regeneration of muscle tissue, skeletal muscles can't divide by mitosis, right? So there are adjacent cells called satellite cells. So during the development, in the embryo, you will have a lot of cells called the myoblasts, these cells. So these myoblasts differentiate into the skeletal muscles, and they fuse together and form the, so they each have a nucleus. So because they fuse together, they have a lot of nuclei, and this is one cell. And these myoblasts, some of them remain as myoblasts, even in the adult. So these remaining myoblasts, these are called the satellite cells. So when there's damage to the muscle cell, if there is damage to these muscle cells, the satellite cells, they get activated. So when these get activated, they start to divide. again, this process will begin, right? And they will form skeletal muscles. This is effective in small muscle damages, but in large muscle damages, this may not be much effective, right? Because there is a limited amount of satellite cells. Okay. So cardiac muscles, they also have very little regenerative capacity, and usually there are fibroblasts between the cells, and when there is damage to the muscle cells, they are replaced by connective tissue, and this forms scars, right? So in myocardial infarction, in heart attacks, because of reduced blood supply, the cardiac muscles, they get damaged, they die off, right? And then they get replaced by scar tissue. So this scar tissue, they can't function as the normal heart muscle, right? They can't contract. So this will lead to reduced function of the heart, and it will lead to heart failure, right? Heart failure is when there's reduced function of the heart. This is usually not reversible. It will gradually lead to gradually decreasing function of the heart, right? So there are some researches going on about replacing this scar tissue with viable heart tissue, heart muscle cells. So this is in this link you can find a TED talk about it, right? It's pretty interesting. You can watch it later on, right? After the lecture you can watch. I think the lecture will finish early on. So after the lecture you can watch this video. So then when it comes to the smooth muscles, they are capable of mitosis. That is, they can divide, forming similar muscle cells. So when there's injury, they can divide, forming new cells to replace the destroyed cells. So when it comes to smooth muscles, there's not much of a problem. Okay. So we have come to the end of the lecture, right? So in summary, we have learned about the types of muscles. Then when it comes to skeletal muscles, we learned about the organization, contraction, innervation, and the types. And about the cardiac muscles, again, the organization, contraction, and a little bit about the conduction system. And in the smooth muscles, again, the organization, contraction, and innervation. And finally, we learned about the regeneration of muscle tissue. So these are the references I used: Junqueira's histology and the Veter's histology. If you have any questions, you can ask now.